Two sisters spat into plastic tubes and mailed their samples to 5 different DNA testing companies. The same 5 companies, the same 2 sisters. These 2 women are identical twins. They came from a single fertilized egg that split in 2. Every letter of their genetic code, all 3 billion of them, is, for all practical purposes, the same. And when the results came back, not one of the 5 companies gave them the same answer. Not one. Most people who take an ancestry test eventually have a version of this experience, in a smaller way. You compare with your brother or your sister, and the numbers don't line up. You are 31% Irish, and they are 19. You have a small slice of something they don't have at all, Scandinavian or Iberian or Ashkenazi. And for a moment, standing there in the kitchen, a strange thought crosses both of your minds. Are we sure we're full siblings? In the next 20 minutes, we're going to answer that question completely, and then take it somewhere most people never get to. Because the ordinary version, why your sister's percentages differ from yours, has a real, elegant, biological explanation. It's true, it's satisfying, and by the end of the next few minutes, you'll understand it well enough to explain it to your family at Christmas. And then we're going to look at the twins again. Because for identical twins, that biological explanation does not work. It cannot work. And what's left when it stops working tells you something about your own results that changes how you read them for the rest of your life. Start with what you were taught, because it's almost right. You inherit half your DNA from your mother and half from your father. That much is true. And it does not change. 50% from each, every one of us. But there's a second half of that sentence that almost nobody hears, and it is where the entire mystery lives. You inherit half of each parent's DNA, but which half is decided at random. Not chosen, not balanced, not fair. Random. Here's what actually happens in the quietest and most consequential lottery of your life. Your mother carries 2 copies of every chromosome. One she got from her mother, one from her father. When her body makes an egg cell, it does not simply hand one of those copies along. First, it does something remarkable. The 2 copies line up alongside each other and physically swap pieces. Sections cross over and trade places, stitching together a brand new chromosome that is part her mother's and part her father's, spliced at points that were not planned and will never be repeated. Geneticists call this recombination. It happens in every egg and every sperm cell, every time in every generation of every sexually reproducing species on Earth. It is the reason no 2 siblings have ever been identical, going back as far as there have been siblings. So the egg that became you carried one particular shuffle of your mother's ancestry. The egg that became your sister carried a different one. Same 2 parents, same available deck, 2 different hands. Now put a number on it. On average, full siblings share about half of their DNA. But on average is doing an enormous amount of work in that sentence. In reality, the figure moves. Some sibling pairs share meaningfully more, some meaningfully less. You and your sister are not 50% the same. You are approximately 50% the same, and approximately is where family arguments are born. Now think about what that does to a great-grandparent. Say one of your 8 great-grandparents was Italian and the rest were not. In theory, that ancestor accounts for 1/8 of you, around 12.5%. But you didn't inherit a tidy 12.5% slice with her name on it. You inherited whatever fragments of her happened to survive 3 consecutive random shuffles on the way down to you. You might have inherited a generous share. Your sister might have inherited noticeably less. And that difference is not an error. It is not the company being careless. It is the ordinary, correct, beautiful consequence of how inheritance actually works. So when your brother's results come back with a slice you don't have, Scandinavian, Iberian, something unexpected, the answer is almost always simply this: He got a piece of an ancestor that you didn't. He isn't more Irish than you. He's carrying more surviving fragments of the same Irish person. And that explanation is true and complete. And it accounts for almost every mismatched pie chart in almost every family on Earth. It just cannot account for the two sisters we started with, which brings us back to the two sisters and their 5 envelopes. Because everything we have just established, the shuffle, the lottery, the random half, depends entirely on one condition. It only explains differences between people whose DNA is different. Charlesy and Carly Agro are identical twins. They are monozygotic, which means they began as a single fertilized egg that divided. There was no second shuffle. There was no different hand. Whatever recombination happened once, before the split, and both sisters carry the result of it. Their DNA is, for the purposes of an ancestry test, the same DNA. So when 5 companies return 5 sets of mismatched percentages for these 2 women, recombination has nothing to say about it. The biological explanation we just spent 10 minutes building does not apply here at all. Something else is producing that difference, and it isn't in their bodies. The test was run by the Canadian Consumer Investigation Program, Marketplace. Charlesy Agro, one of the twins, was the host. In the spring of 2018, she and her sister Carly sent their samples to 5 of the largest names in consumer genetics: Ancestry DNA, MyHeritage, 23, and Me, Family Tree DNA, and Living DNA. The results were published in January of 2019. Every company returned broadly similar portraits of the sisters. That part is worth saying clearly, because this is not a story about tests being nonsense. The overall picture, the continents, the major regions, held up. It was in the detail that things came apart. One company put Charlesy's Eastern European ancestry at 28%. It put her identical sisters at 24.7. At 23 and Me, Charlesy came back with almost 10% less broadly European ancestry than Carly. And Charlesy was assigned 2.6% French and German ancestry, a category her identical twin was not given at all. Read that once more. One sister has a French and German heritage, the other, carrying the same genome, does not. Both of those statements came from the same laboratory, about the same DNA, in the same week. The results were reviewed by a team of computational biologists at Yale University, and their conclusion is the quiet center of this whole story. They did not find contamination. They did not find a mix-up in the mail, or a mislabeled tube, or a technician having a bad morning. What they pointed to was the algorithms. Each company runs its own calculation, its own method of deciding what your DNA most resembles. And as the Yale team put it, the way those calculations are run is different. The samples were the same. The mathematics were not. To understand why that matters so much, you have to know what one of those percentages actually is, and it is not what the pie chart implies. When a company tells you that you are 31% Irish, it has not found an Irish gene. There is no such thing. There is no marker in your body stamped with a country, because countries are a few hundred years old, and your DNA is considerably older than that. What the company has actually done is a comparison. It holds a reference panel, a collection of DNA samples from living people who, as far as the records show, have deep roots in one place. People whose grandparents and great-grandparents all came from the same region. Then it takes your genome, walks along it in segments, and asks over and over, of all the reference groups I have, which one does this stretch look most like? Add up all those small judgments, and you get a percentage, which means your ethnicity estimate is not a measurement of your body. It is a statement about resemblance. How much of your DNA looks like the DNA of a particular group of modern people the company happened to collect? And that is why 2 identical genomes can produce two different answers, change the reference panel, change the segment length, change the statistical thresholds, and the same DNA gets described in slightly different words. The sisters didn't differ. The descriptions of them did. It would be easy to end this by saying the tests are worthless. That would be satisfying, and it would also be wrong, and this channel is not in the business of trading accuracy for a stronger ending. The companies are not inventing your results. The overall picture, the broad continental ancestry, the major regions, is generally reliable, and it held for the twins. The science underneath is real. The problem is the presentation. A number like 31.4% looks like a measurement. It looks like a fact about your body delivered to a decimal place, the way a scale gives you your weight. So we read it as 1. But it is an estimate with a margin of uncertainty around it, and that margin usually is not printed on the pie chart. The estimates get revised as the reference panels grow, which is why your own percentages have probably changed since you first opened the email, and why they will change again. Your ancestry didn't move. The map they were measuring you against got redrawn. So what can you actually trust? Here's the thing almost nobody explains. Your DNA report is not one result. It is 5 completely different kinds of information, stacked on the same page, presented in the same font, as though they were all equally certain. They are not. Some of them will change again. Some of them can never change. Let's go through them, from the softest to the hardest. This is the bottom of the report and the top of your attention. The narrow slices: 2.6% French and German, 4% Sardinian. The single line that made you sit up. This is the layer that moved between 2 identical sisters. It is produced by asking which reference group a short stretch of your DNA most resembles, and when 2 groups genuinely resemble each other, as neighboring European populations do after 1,000 years of intermarriage. That judgment can tip either way on very little. Treat anything under about 5% as a whisper, not a sentence. It might be a real trace of a real ancestor. It might be the algorithm rounding an ambiguity in one direction. On its own, it is not enough to rewrite your family history. And it is certainly not enough to claim a heritage. Move up a level, and the ground gets much firmer. Broadly, Northern European, West African, East Asian, the large blocks. These held for the twins. They hold in general. When a test tells you the bulk of your ancestry traces to a continent or a major region, that is a well-supported statement, because the differences between distant populations are large, old, and easy for an algorithm to see. The signal is loud enough that no reasonable change in method will erase it. So read the big blocks as broadly true, and the fine slices as opinion. The pie chart gives them equal visual weight. They do not deserve it. Now we leave estimates behind entirely. When the test reports that you and another person share a stretch of DNA, measured in units called centimorgans, it is not comparing you to a panel of strangers from a database. It is comparing your genome directly against another individual genome, letter by letter, and finding the places where you match. That is measurement, not inference. There is no algorithmic judgment call about which population you resemble. Either you and that person share a physical piece of chromosome, or you do not. This is why segment data is the currency serious genealogists actually work in. It is also why a family-shaking discovery, the cousin nobody mentioned, the parent who turns out not to be a parent, can be so devastatingly reliable while the percentages beside it are still being revised. Built directly on those segments is your match list, and it is the most underappreciated thing you own. If the test places someone in your close relative range, that relationship is real, not estimated, not a resemblance to a reference group. A measured quantity of shared DNA between 2 living people. Think about what that means. The percentages told those twin sisters two different stories about who their ancestors were. But if either of them had a first cousin in that match list, both would have found the same cousin with the same shared segments at both companies. The soft layer disagreed. The hard layer could not. Your match list is also the only part of the report that can genuinely answer a question. A percentage can suggest your family story has some truth in it. A documented cousin sharing a documented segment descended from the same documented ancestor. That can prove it. And then at the very bottom of the report, usually behind a link almost nobody clicks, is the oldest and most permanent thing you carry. Your haplogroups are deep ancestral lineages. One traced through your mother's mother's mother, back without interruption. And if you are male, one through your father's father's father, along the Y chromosome. They are defined by specific mutations, tiny, harmless copying errors that happened once in one human being thousands of years ago and were passed to every single descendant of that person ever since. You either carry that mutation or you do not. There is no percentage. There is no confidence interval, which means this. When the company updates its reference panels next year and your percentages shift again, and they will, your haplogroup will not move. It did not move when they revised the database last time. It will be the same code on the day you die, and the same code for your grandchildren. And it was already the same code when your ancestors were living somewhere you have never heard of, tens of thousands of years before anyone drew a border. The percentages are the softest number on the entire report. They are also, almost always, the only number anyone looks at. Knowing the layers is one thing. Here is how it applies to the 4 situations that actually send people looking for answers. This is the common one. And now you know it is not a red flag. It is recombination. You each received a different random half of the same 2 people. And the great-grandparents who survived that shuffle in you are not the same ones who survived in them. If the difference is in the fine slices, expect it. If one of you shows a small region, the other lacks entirely, that is still ordinary. A single ancestor, 5 or 6 generations back, can genuinely appear in one sibling's DNA and be completely absent from another's. What would actually be meaningful is a difference in the large blocks, not a few percent, but a major region present in one sibling and missing in the other. That is rare, and that is when you stop reading percentages and go look at the match list instead. Nothing happened to you. The reference panels grew. The company added more samples from more places, reran the comparison, and your genome was redescribed against a better map. Your Irish did not drain out of you overnight. The estimate of how much your DNA resembles a modern Irish reference group was revised. Expect this to keep happening. The fine slices will keep moving. The big blocks will mostly stay put. Your haplogroup will not move at all. Be careful and be patient here in both directions. A small unexpected percentage is not proof of a hidden ancestor, and the absence of an expected percentage is not proof your family lied. Both of those conclusions are drawn every day, and both are drawn too fast. If you want to test a family story properly, you do not do it with the pie chart. You do it with the hard layers. You look for matches who descend from the ancestor in question, and you look at whether you share real segments with them. A story that is true will usually leave relatives behind, not just a percentage. Then you are looking at the most ordinary thing in genetics, dressed up as a revelation. That is not a secret coming out. That is recombination, doing exactly what it has done in every family that has ever existed. You each carry a different sample of the same ancestors. Neither of you got the whole of them. Nobody ever does. If you have compared results with a sibling, the comments below are worth adding to, not the argument. The numbers. How large was the gap, and was it in the big blocks or the small slices? A few hundred of those gathered in one place would say more about how far these estimates really move than most of what has been published on it. There is something worth sitting with in all of this. We took these tests hoping for a verdict, a number that would tell us, finally and precisely, who we came from. And what the two sisters accidentally proved is that the verdict was never that precise, that the percentages are our best current description of an inheritance far older and far messier than any category we have invented for it. Your ancestors were not 31% anything. They were people who moved and mixed and crossed borders that hadn't been drawn yet, and their descendants are still being sorted into boxes those people would not have recognized. The estimate will keep changing. The relatives it finds, you will not. And the deep lineage written into your mitochondria was set down long before anyone thought to name a country. What we do know for certain is that two sisters spat into two tubes and got two different histories back, and both of those histories belonged to the same person.